A very good afternoon to all. Quickly answer through poll. My audio is clear and my screen is visible. So today we are going to discuss sexual reproduction in flowering plants. As you all know that all the flowering plants, they belong to angiosperms. And a flower could be a bisexual flower or a unisexual flower. Bisexual flower which have both androecium and gynoecium. And androecium is the male reproductive part and gynoecium is the female reproductive part. As you can see in the image, it's a bisexual flower. It contains the androecium and gynoecium. Unisexual flowers are those which either have a androecium or gynoecium. Androecium is a wall of stamens and gynoecium is a wall of carpels. Remember their unit. Unit of androecium is stamen. Unit of gynoecium is carpel or pistil. Unit of calyx is sepal. Unit of corolla is petal. Okay, remember this. Wall of stamen is known as androecium, and stamen is consists of two parts, anther and filament. Anther is a bilobe structure. This is a lobe. This is lobe 1 and 2. So it is a bilobe structure. And this part is the filament. This is the distal part of the filament. And this is a proximal part of the filament. We are calling proximal and distal with respect to the thalamus. The end which is closest to the thalamus is known as proximal, and the end of the filament which is away from the thalamus is known as distal. So the proximal end of the filament is attached to the thalamus or petal. And distal part of the filament is attached to the anther. Remember this. Distal part of the filament is attached to the anther. Anther is a bilobe structure and diathecous. Diathecous means theca is a chamber. So in one lobe, there are two theca are present. This is a lobe 1, lobe 2. It has two theca. These are the theca. So in one lobe, there are two theca. So it is diathecous. It is tetrasporangiate because there are 4 microsporangium are present. So it is tetrasporangiate. It is tetragonal in shape. For example, if I divide this anther, we get 4-sided structure. So it is tetragonal in shape. And there's a longitudinal groove which runs lengthwise and it is dividing the two or separating the theca. Moving next is microsporangia. It consists of, the anther consists of 4 layers. The outer is epidermis, then there's endothecium, which also contains, which is, okay, let me draw it here. First one is epidermis. Second one is endothecium, which has radial thickening. Inner to the endothecium are middle layer, which are 2 to 3 layers. And the function of this epidermis, endothecium, and the middle layer is protective in nature. And second, they help in dehiscence of anther to release the pollen. And there's a fourth layer, is the tapetum, which is the innermost layer, and the tapetum is nutrition in nature. It is nutritive in nature. It provides the nourishment to the sporogenous tissue. And the sporogenous tissue, it is a compactly arranged homogeneous cells which occupy the center of each microsporangium. This is the sporogenous tissue. And the cells of the sporogenous tissue are known as a microspore mother cell. It is diploid in nature. So microspore mother cell will undergo meiosis and will produce the 4 haploid microspores. So this is a microsporocyte or the microspore mother cell. This microspore mother cell will undergo meiosis 1 and meiosis 2 and result in the haploid microspore. These microspores, when they are in tetrad condition, they are surrounded by a wall which is of callose wall. Remember this. Around the tetrad of microspore, there's a callose wall is present or the callose layer is present. And these microspores, they undergo mitotic division to produce a mature pollen grain. Let me show you in this diagram. The pollen grain, it has 2 layers. Outer is exine and the inner is intine. The exine layer, it consists of sporopollenin. It is made up of sporopollenin, and sporopollenin is the hardest substance. It cannot be easily degraded by the enzyme, and it is also resistant to strong acid and base, because of which it can withstand the high acidic conditions and the high basic conditions, and it is also cannot be degraded by the enzyme easily. So that is why the pollen, they are preserved as fossils. And this exine, it is discontinuous. You can see this, it is discontinuous. These are known as germ pore. So these germ pore are present through which the pollen tube will come out. And the inner layer, it is thin in nature and it is made up of cellulose and pectin. And this inner layer, which is known as intine, it is continuous. Now, the mitotic division occurs in pollen and it is divided into 2 cells. There is vegetative cell and generative cell. Vegetative cell is large in size and it consists of abundant food reserve and it has irregularly shaped nucleus. And the generative cell is smaller in size and it is floating in the cytoplasm of the vegetative cell and it has spindle-shaped nucleus. In 60% of the angiosperm, the pollen grain, they are shed at 2 cell stage. Okay. And in 40% of the angiosperm, the pollen grains, they are shed at 3 cell stage, where this generative cell undergo mitosis and it produces 2 male gamete. So now the cell has vegetative nucleus and the 2 male gamete. Pollen grains, they are generally spherical in shape and they're having a diameter of 25 to 50 millimeter. Pollen grains of various species, they cause allergies or chronic respiratory disorder like asthma, the bronchitis, parthenium. It causes the pollen allergy. Please remember this. The pollen, they are rich in nutrient, because of which their pollen tablets and syrup are present in the market that claim to increase the performance of athletes. The pollen viability. Viability is the lifespan. For example, every medicine has a manufacturing date and the expiry date. So this is lifespan. So the pollen viability is highly variable. It depends upon the temperature and the humidity. In species like rice, wheats, the pollen, they lost their viability within 30 minutes. Like there are some members of Rosaceae, Leguminosae, and Solanaceae, the viability can be for months. And we can also store the pollen grains through cryopreservation in the pollen banks for breeding, for crop breeding programs. Next, we are going to discuss is the pistil, megasporangium, and the embryo sac. First, we are going to discuss the gynoecium. Gynoecium is a female reproductive part. If the gynoecium is consist of single pistil or single carpel, then it is known as monocarpellary. Mono means one, carpellary is carpel. If the gynoecium is having more than one pistil, then it is known as multicarpellary. Multi means many, carpellary means carpel. So it having multiple carpels. It can be apocarpus or syncarpus. Apo means free. The carpels, they are free. Syncarpus, that carpels are fused. For example, in this is a condition of paperwork where the gynoecium is multicarpellary and it is syncarpus. Second is the machlea. This is the example of machlea where the gynoecium is multicarpellary and it is apocarpus. You can see that there are many carpels and these carpels, they are free. They are apocarpus in nature. And we know that the pistils consist of three parts. There's a stigma is present on which the pollen will settle or get deposit. Second one is style, which is a long tube. And at the basal, there's an ovary is present. Ovary consists of ovarian cavity, which is known as locule. And in the locule, there's a placenta is present. And the ovule is attached to the placenta with a stalk-like structure known as funicle. So this is a funicle, the stalk-like structure. And the hilum is the junction where the funicle is attached to the ovule. So hilum is that junction where the funicle is attached to the ovule. And the ovule is consist of integument. There's outer integument or the inner integument, which on maturation, when the ovule get fertilized and form the zygote, then the mature ovule, we call it as mature ovule. Now, that time, these outer and the inner integument will form the seed coat. The outer integument will form testa and the inner integument will form tegmen. This nucellus is present and this embryo sac. In wheat, paddy, and rice, there's one ovule in one ovary. So in case of wheat, paddy, and mango, there's one ovule in one ovary and many ovules in many ovary in case of papaya, watermelon, and orchids. Now moving next is megasporangium. Megasporangium, we also refer it by ovule. So now look at the figure. This is the ovule. In the center, this is nucellus. Nucellus is diploid in nature. So one of the cell of the nucellus at the micropylar end get differentiated into megaspore mother cell, which is diploid in nature. And this big megaspore mother cell will undergo meiosis and 4 haploid megaspores are formed. Out of 4, the 3 get degenerated. So 3 haploid megaspore get degenerated, only 1 remain functional. And this functional megaspore will form the embryo sac. We also call it as female gametophyte. Now this embryo sac will undergo 3 mitotic division and form 8 nuclei. So 3 will move to the chalazal end, form the antipodal cell. The 3 will move towards the micropylar end, which consists of 2 synergid and 1 egg. And the 2 remains in the central, which form the central cell, or which contains the 2 polar nuclei. As you can see, this is the structure of the female gametophyte and the embryo sac. At the chalazal end, there's a 3 antipodal cells are present. At the micropylar end, there are 2 synergids are present, and these synergids, they have filiform apparatus which guide the entry of the pollen tube. In the center, there's a central cell is present which consists of 2 polar nuclei. So this is a condition where there's a 7 cell, 1, 2, 3, 4, 5, 6, and 7. So there's a 7 cell, but it is 8 nucleated. Remember this. Very, very important. It is 7 cell, but 8 nucleated. There are 8 nuclei per 7 cell. And the development of this embryo sac is monosporic development, because a single megaspore is lead to the formation of an embryo sac. So the development is monosporic. Moving next is the pollination. Pollination is the transfer of the pollen to the stigma of the flower. So it could be autogamy. Autogamy means self-pollination. In case of the autogamy, the pollen from the anther is transferred to the stigma of the same flower. So that means the pollen transfer is within the flower, within one flower. This is known as autogamy, and autogamy is also referred as self-pollination. This process, this autogamy process, it requires synchronization between the pollen release and the stigma receptivity. So the pollen release and the stigma the receptivity of the stigma should take place at one time. It should be synchronized. Then only the autogamy will take place, or self-pollination will take place. There are clistogamous flowers, which are those flowers which does not open at all, that remain closed. In these flowers, the pollination takes place because the pollen and the stigma, the anther of the stigma, they are present in close proximity of each other. So the pollination will take place, but pollinators are not involved in this case. So please remember, there's an absence of pollinator. These species, they also produce chasmogamous flowers. Chasmogamous flowers where the anther and the stigma are exposed for cross-pollination. Remember the name of the species, they are Viola, Oxalis, and Commelina. Moving next is zytomogamy. In zytomogamy, the pollen are transferred from the flower to the stigma of the another flower, but of the same plant. Remember, they are of both the flower, they are of the same plant. So the pollen from this flower is transferred to the stigma of this flower, another flower, but of the same plant. So it is zytomogamy. In this case, it is functionally cross-pollination. But why it is functionally cross-pollination? Because it is involving the pollinating agent. But it is genetically autogamy. Genetically, it is autogamy because the flower belongs to the same plant. Okay. In xenogamy, the different plants are involved. That means the pollen are transferred to the stigma of the different flower of the different plant. So xenogamy is also referred as cross-pollination. Moving next is the pollinating agent. It could be abiotic agent like wind and water. And the pollen coming in contact with stigma is a chance factor in both wind and water pollination. Remember this, it's a chance factor. A flower produces a pollen grain in a very large amount because when these pollens, they are carried by this abiotic agent like wind and water, a large amount of pollen get lost. So to compensate that, the pollen, they are produced in a very large amount as compared to the number of ovules. For wind pollination, first condition required is the pollen should be light, should be non-sticky, and the stigma should be feathery, well exposed, and should be feathery so that the stigma can easily trap the airborne pollen. Wind pollination, it is quite common in grasses. Water pollination, it is rare. Okay, and in majority of the aquatic plants like water hyacinth, water lily, and in which the flowers, they emerges above the water. So if this is water, their flower will come out of the water for the pollination. In Vallisneria, this is a diagram of water pollination in the species Vallisneria, where the flower, female flower, it's coming out on the surface of the water for the pollination. So this type of pollination is known as epihydrophily. In sea grasses or Zostera, it takes place below the water, which is hypohydrophily. Epi means above. In Vallisneria, it is above, and in Zostera, hypo means below. So it is in sea grasses or Zostera, the water pollination is taking place below the water. And in most of the water pollinated species, the pollen grains are protected from wetting by mucilaginous covering. Because if the pollen, they get wet, they will become heavy and they will settle down. So it will affect the water pollination. So in most of the water pollinated species, the pollen grains, they are covered by the mucilaginous covering to protect the pollen from getting wet. Next is the biotic agent, which includes insects like bees, butterflies, flies, beetle, wasps, ants, moths, birds, and bats. Bees, remember, bees are the dominant pollinating agents. Some primates like lemur, arboreal tree-dwelling, rodents, even reptiles are also pollinators in some species. In the image, you can see there's a pollination, biotic pollination is taking place. So the insect, the flower, it is getting the pollination. So the insect is helping the flower by pollination, and the insect in return is getting the flower reward. Okay, flower reward could be a nectar, and it is a safe place to lay the egg. For example, many species like Amorphophallus, yucca, or the fig plant, in these, the insect lay their egg in the flower. So the insect, they are helping the plant or the flower by pollination, and in returns, the insect is getting the floral rewards. Insect pollinated flowers, they are large, colorful, pregnant, and rich in nectar to attract the insect. And nectar and the pollen grains are the usual floral rewards. Next one is the outbreeding devices. The flowering plants have developed many species to discourage the self-pollination and to encourage the cross-pollination. First one is that pollen release and the stigma receptivity are not synchronized. Remember, in case of autogamy, the pollen release and the stigma receptivity are synchronized so that the self-pollination can take place. But to promote cross-pollination, the pollen release and stigma receptivity should not be synchronized. Second one is the different position of the anther and the stigma. If the anther and the stigma are present at the different position, then the pollen cannot come in contact with the stigma of the same flower. For example, if the stigma length is larger as compared to the anther, then because of that, the pollen cannot come in contact with the stigma of the same flower. Self-incompatibility. Self-incompatibility is the genetic mechanism by which the pollen from the same flower cannot germinate on the stigma of the same flower. For example, as you can see in this image, the stigma having a molecular configuration as S1 and S2. So the pollen which are having S2 and the S1 molecular configuration, they won't be able to germinate on the same stigma. So there's a rejection is taking place at a molecular level. This is what we call self-incompatibility. And the pollens which are having a different molecular configuration, they're able to germinate and they produce the pollen tube. So that means these pollens are compatible, and the compatible pollen will able to germinate and will produce the pollen tube, and this pollen tube will reach to the one of the synergid of the embryo sac. And the production of unisexual flower, which is a dioecious flower. If the plant is dioecious or producing a unisexual flower, then there is a chances that this plant will show cross-pollination. In case of castor and maize, which is a monoecious means bisexual, the autogamy is prevented but not xerotonogamy. In papaya, which is dioecious, both autogamy and xenotogamy are prevented. Remember, this is very important. Moving next is the pollen-pistil interaction. Pollen-pistil interaction is the interaction between the pollen grains and the pistil, where the pistil will either reject or accept the pollen grains. It depends upon the compatibility of the pollen grain. If the pollen grain is compatible, that pollen grain will able to germinate and will produce the pollen tube, and this pollen tube will enter from the micropylar and will enter to one of the synergid of the embryo sac, and the synergid, they contain the filiform apparatus which guide the entry of the pollen tube. And this pollen tube, they contains the two male gametes. So all these events from the pollen deposition on the stigma until the pollen tube enter the ovule are together referred as a pollen-pistil interaction. Next one is artificial hybridization. In artificial hybridization, desire pollen are used for pollination, and the stigma is protected from contamination from the unwanted pollen by emasculation and bagging. This is a bisexual flower. We are removing the anther. Removal of anther is known as emasculation. And after this, after the removal of the anther, the flower is bagged to avoid the contamination from the unwanted pollen. So we bag the flower. Next we do is the dusting of the desired pollen on the surface of the stigma. And again, we rebag the flower for the fruit development. So in artificial hybridization, we are promoting cross-pollination or the pollination with the desired pollen grains. Moving next is double fertilization, very important. So the pollen tube will enter to one of the synergid of the embryo sac, and these synergid, they contain filiform apparatus which guide the entry of the pollen tube. Now the pollen tube will release the male gamete. One male gamete will fuse with the egg and form the zygote. This process is known as syngamy. And one of the gamete will fuse with the central cell and form the endosperm, which is triploid in nature. Remember, the ploidy zygote is diploid and the endosperm is triploid in nature. And the endosperm is formed by triple fusion. Because the central cell has two polar nuclei and the one male gamete, which is released by the pollen tube. So there's a triple fusion is taking place, which result in the formation of the endosperm, and endosperm has a ploidy of 3. Main function of the endosperm is to provide the nourishment to the developing embryo. Please remember that double fertilization is the combination of both syngamy and the triple fusion. Next one is post-fertilization event. So endosperm, it is larger in size. It has a reserve food material to provide the nourishment to the developing embryo. So the zygote will start dividing or converted into embryo when there's a sufficient amount of endosperm is formed. Okay, so first the development of endosperm will take place, then the zygote undergo mitosis to form embryo, because the embryo requires certain amount of endosperm to provide the nourishment. So this embryo can receive the nourishment from the endosperm. So most common type of endosperm is a nuclear type, where the primary endosperm nucleus undergoes successive nuclear division to give free nuclei. We call this condition as syncytium, multinucleate condition. Example is coconut water. And the white kernel is known as the cellular endosperm. When the zygote undergo mitotic division, it forms the embryo. And the early stages of embryo development, we call it as embryogeny. First, zygote will form proembryo, then it will convert it to globular embryo, then heart-shaped embryo, torpedo embryo, and then the mature embryo. Okay? Now the embryo consists of cotyledon and the embryonal axis. So this is a diagram of a dicot seed. The example of dicot embryo because it's having a 2 cotyledons. As you know that dicot has 2 cotyledons, dicot seed has 2 cotyledons, and monocot seed they have only 1 cotyledon. So in this diagram, there's 2 cotyledon, cotyledon number 1 and cotyledon number 2. And this is the embryonal axis is present. And this point is the point of attachment where the 2 cotyledons are attached. So the part of the embryonal axis, which is above the point of attachment where the cotyledons are attached, this part of embryonal axis is known as epicotyle. And the part of embryonal axis, which is below the point of attachment of the cotyledon, this is known as hypocotyle. So epicotyle will terminate into pimule, which forms the shoot, and the hypocotyle will terminate into radicle, which forms the root. And in case of grasses, the embryo have a single cotyledon, which is known as scutellum. Remember this name, that the embryo of monocot has 1 cotyledon and its name is scutellum in grass family. And the radicle and the root cap, they are enclosed by the undifferentiated sheath, which is known as coleorhiza. And epicotyle has a shoot apex and few leaf primordia, which are enclosed by a undifferentiated sheath, which is known as the coleoptile. So main function of coleorhiza is to protect the radicle, and main function of coleoptile is to protect the epicotyle. Next one is seed. Mature ovule or the fertilized ovule is known as seed. It is the final product of sexual reproduction and it is formed inside the fruit. So when the ovule gets fertilized or the mature ovule is known as seed. And the mature ovary is known as fruit. And as we know that the ovule is attached to the placenta with the stalk-like structure, which is known as funicle. So the point or the junction where the funicle is attached to the ovule is known as hilum. So when the ovule get mature or get fertilized, the ovule get detached from the funicle. So when they get detached from the funicle, it leaves a scar, which is this hilum. And there's a opening is present, which is known as micropyle, through which the seed absorb water, nourishment, and get oxygen. Seeds generally have two things. One is the seed coat and the embryo. And the embryo is consist of cotyledon and the embryonal axis. Seed coat, which are formed when the, that ovule has, when it is in the young stage, it has the outer integument and the inner integument. So when the ovule gets fertilized or mature, these outer and the inner integument, they form the seed coat. Outer integument will form the outer seed coat, which is known as testa, and the inner integument will form the inner seed coat, which is known as tegmen. And there's two types of seeds are present. There's non-albuminous seed, which is also known as non-endospermous seed, example pea and groundnut. Because in these seeds, the endosperm is completely utilized by the developing embryo. So there's no residual of the endosperms are present in these seeds. So they are known as non-endospermous or non-albuminous seed. Albuminous seeds are those seeds which have endosperm, like wheat, maize, barley, castor, and coconut. They all have endosperm. And the albuminous seeds are mostly found in monocot. Monocot had the endospermous seed. And non-endospermous seed or the non-albuminous seed, they're mostly found in dicot. And castor is a dicot plant, but it has the endospermous seed. Remember this. The castor is the exception. It's a dicot plant, but it has the endospermous seed. Black pepper and beet, they have the perispermic seed because they have the in the seed, the persistent nucellus is present, and persistent nucellus is known as perisperm. So the seed in case of black pepper and beet is known as a perispermic seed. There are two types of fruit, true fruit and false fruit. The fruit which are forming from the ovary is known as a true fruit, and the fruit which are forming from the part of the flower other than ovary are known as a false fruit. For example, the apple, strawberry, cashew, they all are the false fruit. The apple and strawberry, they are formed from the thalamus. Instead of ovary, they are formed from the thalamus. So they are false fruit. Parthenocarpic fruit are those fruit which are formed without fertilization. Example, banana. Without fertilization, that means there's no formation of seeds. So the banana is a seedless fruit. So parthenocarpic fruit are those fruit which are developed without fertilization. Now, just like the pollen have viability, similarly the seeds, they also have viability. Lupinus arcticus, it was excavated from the Arctic region, and the seed of this plant has a viability of 10,000 years. So the seed of this plant has a lifespan of 10,000 years. Pinus dactylifera, which is also commonly known as date palm, this plant has a viability. The seed of this plant has a viability of 2,000 years. Next one is apomixis. Apo means without and mixing means fertilization. So the formation of seed without fertilization is known as apomixis. Mostly the species of Asteraceae and grasses, they show apomixis. This is the ovule. Inside the ovule is a nucellus. This nucellus, one cell of the nucellus differentiated and formed the megaspore mother cell. This megaspore mother cell will undergo meiosis and will produce 4 haploid megaspore. Out of 4, 3 get degenerated and only 1 is functional. The one which is functional will form embryo sac or the female gametophyte. Embryo sac has egg. So when the male gamete from the pollen through the pollen tube enter into the embryo sac and the male gamete fuses with the egg, it will result in the formation of zygote. This zygote will undergo mitotic division and form embryo. An embryo is a part of seed. Right? So this is the general process. But if this cells of the nucellus, instead of forming the megaspore mother cells, it directly forming the embryo. And this whole process is not taking place. Because this process includes reduction and fertilization. Both this process is not taking place and the nucellus cells are directly forming the embryo, then the seed is formed is known as an apomictic seed. And the process is known as apomixis. Similarly, in some species, the megaspore mother cell directly forms the diploid embryo sac. And this diploid embryo sac will have diploid egg. And this diploid egg will directly form the embryo. In this case also, there's no reduction division, there's no fertilization. So here the seed form is also the apomictic seed. Okay? And when, remember, in case of citrus and mango, this nucellus cell is directly forming the embryo. Okay? And if more than one nucellus cell is forming the embryo, then the condition, it results in a condition which is known as polyembryony, which you can observe in case of citrus and mango. Now let's discuss the question. Question number 1 says double fertilization is, option 1 is a fusion of two male gametes of a pollen tube with two different eggs. Option 2 is fusion of one male gamete with two polar nuclei. Option 3 is fusion of two male gamete with one egg. Fourth is syngamy and triple fusion. Quickly answer in the chat box what you think what will be the correct option for this question. Very good, MS Gauri. So double fertilization. Remember, this double fertilization is a unique event of flowering plants. And all the flowering plants, they belong to angiosperms. Right? In double fertilization, there's two male gametes. One fuse with the egg and forms zygote, which is known as syngamy. Other fuses with the central cell, which contains two polar nuclei, and form primary endosperm nucleus. And the process is triple fusion. And this syngamy and triple fusion collectively known as double fertilization. So correct option would be 4, syngamy and triple fusion. Option 1 says fusion of two male gamete of a pollen tube with two different eggs. No. Fusion of one male gamete with two polar nuclei. No, because double fertilization includes both syngamy and triple fusion. Fusion of two male gamete with one egg. No. So the correct option is option number 4, which includes syngamy and triple fusion, which are collectively result in a condition called double fertilization. Next one is double fertilization is exhibited by A is algae, B is fungi, third is angiosperm, and fourth says gymnosperm. What do you think? What will be the correct option for this question? Very good, MS Gauri. Just now I told you that double fertilization is a unique feature of angiosperm. So double fertilization is exhibited by angiosperm, and angiosperm includes all the flowering plants. Moving next is the filiform apparatus is characteristic feature of. Option A is synergid, option 2 is generative cell, option 3 is nucellar embryo, option 4 is aileron cell. What do you think? What will be the correct option for this question? Very good, Ms. Gauri. So filiform apparatus is a characteristic feature of synergid. This is the embryo sac or the female gametophyte. This portion is the chalazal end. Chalazal end consists of 3 cells which are antipodal. In the center, there's a central cell is present, which contains 2 polar nuclei. At the micropylar end, there are 2 synergid and 1 egg cell. And these synergid, they have the filiform apparatus. And function of this filiform apparatus is to guide the entry of the pollen tube. Option 2 is generative cell. When this microspore undergo mitosis, two cells are formed. One is the vegetative cell, which is larger in size and has irregularly shaped nucleus. Second one is the generative cell, which is smaller in size and it is floating in the cytoplasm of the vegetative cell. And this generative cell will further undergo mitosis and will produce the 2 male gamete. Nucellar embryo, this is a case of apomixis when the nucellar cells, they without fertilization, they directly forming the embryo. Aileron cells, these are found in the monocot, see where the endosperm is separated from the embryo. In case of maize seed, remember there the endosperm is separated from the embryo by the proteinaceous layer, which is known as the aileron layer. Like this. This is the embryo, this is the endosperm, and both are separated by the proteinaceous covering of the endosperm, which is known as the aileron layer. So, our question is asking about the filiform apparatus, which is a characteristic feature of synergids. So, the correct option is 1. Moving next. In which of the following both autogamy and xerotonogamy are prevented? 1 is wheat, 2 is papaya, 3rd castor, 4th maize. What do you think? What will be the correct option for this question? Very good, Ms. Gauri. So both autogamy and xenotogamy are prevented in case of dioecious plant. In case of dioecious, both autogamy in case of monoecious, autogamy is prevented, but not xytonogamy. So here, option 1 says wheat. Wheat is monoecious. Castor monoecious. Only papaya is dioecious. Remember this. So correct option is 2, papaya. wheat, castor, maize, they all are monoecious. And the question is asking in which of the following both orthogamy and zootonogamy are prevented. That means they're asking about the dioecious. Only papaya is dioecious. Please remember the example. Remember all the examples and diagram, their labeling, these are very important. Question 5 says, is some plants, the female gamete develop into embryo without fertilization? This phenomena is known as. Option A is autogamy. Option 2 is parthenocarpy. Option 3 is syngamy. Option 4 is parthenogenesis. What do you think? What will be the correct option for this question? No, it's not 2. The question is asking the female gamete develops into the embryo without fertilization. This phenomena is known as parthenogenesis. Parthenogenesis, when the female gamete, they develop into the embryo without fertilization. Parthenocarpy is the development of fruit without fertilization. Syngamy, when the male gamete fuse with the female gamete and they form zygote. And autogamy, it is this self-pollination. So the correct option is option number 4. I hope it is clear now, Ms. Gauri. Moving next. In which of the following may require pollinators but is genetically similar to autogamy? Option 1 is xenogamy. Option 2 is apogamy. Option 3 is cleistogamy. Option 4 is zootonogamy. Quickly answer what you think, what will be the correct option for this question. Question is asking which one of the following may require pollinators but is genetically similar to autogamy. Remember, zootonogamy, it requires pollinator. It is functionally cross-pollination because different flowers are involved, but genetically similar to self-pollination or autogamy because both the flower, they belong to the same plant. That is why the correct option is zootonogamy, because which question is asking which of the following requires pollinator. Zootonogamy requires pollinator, but is genetically similar to autogamy. Very good, Ms. Gauri. In zeogamy is the cross-pollination. Cleistogamy, when the flower does not open. And in cleistogamy, autogamy will take place, and apogamy is the formation of embryo without fertilization. Moving next is which of the following set represent the correct labeling of A, B, C, and D with respect to the given diagram. Please remember the diagram which are given in NCERT and also their respective labelings. They are very important. Please remember this. So this is the LS of castor seed. And castor is a dicot. An exception because it has endosperm. Because generally the dicot seed, they are non-endospermic seed. But the exception is castor. Similarly, monocots, they are non-endospermous seeds. And they also have an exception which is orchid. So in monocot, the exception is orchid because the orchid, they does not have endosperm. Okay. So here the castor is a dicot seed, but it has endosperm. So A is the seed coat. P is cotyledon, C is endosperm, and D is hypocotyle. So now let's match. A is seed coat, B is cotyledon, C is endosperm, and D is hypocotyle. So the correct option is option number 4. I hope it is clear to everyone. Please remember all, I'm telling again and again because these are very important. Please remember all the exception. Please remember all the examples and labelings, diagrams and their labelings. These are very important. So that's all for today. We are winding up the class. Thank you all for your participation. I hope this lecture was informative for you. Thank you all. See you in the next class and have a nice day.